For those of you I have not had the pleasure to meet and may not be familiar with solid biosciences, I would love to take a few minutes to share our story and ultimately the reason for Solid's existence. I will then hand it over to Joel Schneider, our Chief Technology Officer, and Cathryn Clarey, our Chief Medical Officer. My son Etani was diagnosed in October 2012. From the moment we received this news, our lives were forever changed. We left our careers and we moved from London to Boston to start a company to not only save our son's life, but all those living with Duchenne. My husband, Inan, and I, together with some extraordinary individuals, launched Solid in 2014. We are a biotech company focused solely on developing treatments for Duchenne muscular dystrophy. We have employed and partnered with the best and brightest experts in Duchenne and gene therapy. We set lofty goals from the beginning to develop transformative therapies that will improve the lives of all patients living with Duchenne, regardless of age, mutation, or disease progression. And to tackle our goals, we live by three core principles. First, we are patient-led. The patient voice is woven into the fabric of everything that we do. We lead with purpose and integrity through listening, learning, and supporting the patient community. We go into every single day with one goal: to improve patients' lives. Second, honesty and transparency. The patient community is a vital stakeholder. Through everything we do as a company, we aim to do as authentically and openly as possible. We believe success is tied directly to the understanding and support of the community behind us. Third, scientific rigor. We're confident that we have the right team in place to bring meaningful therapies to the Duchenne community. We will not stop until we've achieved our goal to advance transformative treatments for those we love and serve. With that, I would like to turn it over to Joel Schneider to provide more information on our lead gene therapy candidate, SGT-001.
Thanks, Annie. As Annie mentioned earlier, my name is Joel Schneider, and I'm the Chief Technology Officer at Solid. First, I'd like to start with a bit of background on our gene therapy candidate called SGT-001. SGT-001 is an investigational gene transfer, a type of gene therapy, of a smaller but functional version of the dystrophin gene called microdystrophin. SGT-001 is delivered systemically, enabling it to enter cardiac and skeletal muscles. The SGT-001 microdystrophin is a differentiated construct based on more than 30 years of research conducted by our advisors, Jeff Chamberlain and Dongsheng Duan. It is the only microdystrophin currently in clinical development that's capable of restoring production of nitric oxide, or nNOS, in the muscle, which is important for proper muscle oxygenation and muscle strength. As many of you know, our clinical trial, called IGNITE DMD, was recently lifted from clinical hold. As part of our commitment to continuously improving our manufacturing processes, we have implemented and shared with the FDA manufacturing changes that remove the majority of empty viral capsids, allowing us to achieve our therapeutic dose target with fewer viral particles. The reduction in the total number of viral particles delivered to each patient is intended to support safe dosing of SGT-001 for the duration of the IGNITE DMD trial. I would now like to pass it over to Cathryn, who will provide an update on the clinical protocol changes in the IGNITE DMD study.
Thank you, Joel. As mentioned, my name is Cathryn Clarey, and I'm the Acting Chief Medical Officer at Solid. For those of you who are not familiar with IGNITE DMD, I'd like to provide a little background on it. IGNITE DMD is our phase 1/2 ascending dose clinical trial, and it's designed to assess the safety and tolerability, as well as the efficacy of intravenous SGT-001 in boys with Duchenne. To date, we've dosed 6 patients at 2 dose levels. 3 patients were dosed at 5E13 vector genomes per kilogram, and then 3 patients were dosed at 2E14 vector genomes per kilogram, which is 4 times higher than the lower dose. We have 2 primary endpoints in the study: safety and microdystrophin expression at 12 months, as well as a number of secondary endpoints. Analyzing our muscle biopsies that were taken 3 months after administration from the 2E14 VIGs per kg dose cohort, remember that's the higher dose cohort, we've demonstrated widespread expression of our microdystrophin throughout the muscle in all 3 patients. We also see, very importantly, the restoration of critical dystrophin-associated proteins. Now I'd like to take a moment to address a question I'm sure you have, which is the recent clinical hold. As you may know, a boy who was dosed in the 2E14 VIGs per kg dose cohort last October had a serious adverse reaction, which led to the clinical hold being placed on IGNITE DMD. In response to the hold, since safety is paramount to our team at Solid, we did a deep dive into all the data collected to date on all 6 patients to understand what happened. We worked with a number of knowledgeable external experts and did develop a deeper understanding of what transpired in this most recent patient. And ultimately, we developed a clinical risk mitigation strategy, which was designed to improve safety for future dosing. As part of sending this to the FDA, and in order to help decrease the risk of serious drug-related adverse events, we amended the protocol of the IGNITE DMD to include the use preventively of 2 anti-complement inhibitors, eculizumab and C1 esterase inhibitor. And we also amended the protocol to include an increase in the prednisone dose in the first month post-dosing. In addition, we're reducing the maximum weight of the next 2 patients dosed to 18 kilograms. This is close to 40 pounds. This reduction, in conjunction with the delivery of fewer viral particles as a result of the manufacturing process improvements, will reduce the total viral load being administered to patients, and we will be able to continue dosing at 2E14 vector genomes per kilogram. It's important to note, and this is very important to us at Solid, that all 6 patients dosed are doing well. All of the SAEs that have been experienced have recovered, and there have been no additional drug-related adverse events up to 30 months post-dosing. Additionally, when we submitted our package to the FDA, we responded to their request for updated safety and functional efficacy data, including the 6-minute walk test and North Star ambulatory assessment for all patients dosed in IGNITE DMD. We look forward to sharing these data at a future medical meeting when appropriate. We are working diligently now to complete all the activities necessary to resume dosing, and we expect the next patient will be dosed in the IGNITE DMD trial in the first quarter of 2021. For any future updates, please visit clinicaltrials.gov, which is updated, and IGNITE DMD clinical trial webpage. Thank you.
Together,
we're solid.
Together, we are solid.
Together, we are solid.